Hello, students. Welcome to my screencast to help you get ready for your first test. I've got 3 of them for you. The first one will be on Unit 1, Experimental Design and the Characteristics of Life. You all did really well on this. You remembered a lot of it from 8th grade, and hopefully it should go well for you. I'm going to give you some questions from the study guide and then give you some of the slides that we went over in class, and just to help you review and things for you to check out. So let's go. From the study guide, can you design an experiment to test which type of music makes a person have the lowest resting heart rate? What is your independent variable, dependent variable, constants, and control? Another question, can you design an experiment to test whether heat affects the enzyme catalase? What is your independent variable, dependent variable, constant, and control? And then number 3, why is it important to have a control in an experiment? So number 2 is a really good one, right? It goes right back to the lab we did in class. And if you can answer all 3 of these, then you really have a good grasp on experimental design. So go ahead and pause the video and see if you can answer them. And now let's go through. Okay, here were your notes that I gave you. The independent variable is whatever we manipulate in an experiment. It's graphed on the x-axis. Dependent variable responds to it. We measure it and we graph it on the y-axis. Constants are things we keep the same from trial to trial. They're also called the control variables. And a control group is a standard for comparison. So these are definitions. These don't really help us unless we can put them into context of a problem. So let's go back to that first one. Does music affect my heart rate? Now I asked you in the study guide, which one gives you the lowest heart rate? But we'll just use here, does music affect my heart rate? So my hypothesis, if I listen to loud, intense music, then my heart rate will rise. That should be a period right there. So the independent variable would be the types of music. You might have you and your brother or sister or your family member sit down and listen to different types of music. Really loud, intense music. You could have classical music. You could have classic rock music, etc. Maybe some traditional Omani music. And each time you'd be measuring their heart rate, right? You'd be checking the dependent variable. You'd be seeing how it affects their heart rate. Now, it's not fair if one time you have the music super loud and the other time it's really soft. The music volume needs to be the same each time. We also need to have the same length of song. We need to have the same type of bass each time. You're trying to minimize the effect of other variables. You only want to test, students, the different music types. We're not testing your Bose speaker versus a computer speaker, etc. You're also going to have a control group. The L, I remember as L for left alone. So it's going to be the group that serves as a standard for comparison. So we're going to check you with no music at all. Maybe music just doesn't really have an effect, or maybe all music raises your heart rate. So the control group serves as our standard for comparison. So I'm hoping through that illustrative example, it helps you to see the steps of experimental design. Here's what we did in class. This was, I believe, the first or second day of school. Which liquid will help break down hydrogen peroxide more quickly? And then I said a liquid that contains living cells more quickly break down hydrogen peroxide. So we had here our independent variable was the type of liquid, and we tested 2 things in class, if you remember the beakers. We tested yeast water and soap water. Well, hopefully now, after our lab, you know that yeast and living organisms contain the enzyme catalase. And catalase helps us to break down hydrogen peroxide. We saw the yeast water really bubble up, whereas the soap water, nothing happened. So the dependent variable, what were we measuring? We were measuring the breakdown of hydrogen peroxide. And how did we measure it? We saw the release of oxygen gas. You saw the bubbles rise up in the test tube. Things that were constant, right? Ask yourself, the constants can also be called control variables. We had the same amount of yeast water in each tube, the same amount of yeast water as soap water, and we gave the same amount of hydrogen peroxide each tube. It wouldn't be fair if we gave one tube, say, 5 milliliters, and the other one 2 milliliters. The control group, we compared it against room temperature water. All right, can you go through and figure out the independent variable, dependent variable, constants, and control? On your test, you're going to have a scenario where you need to identify the independent variable, dependent variable, constant, and control. All right, now we're moving on to the characteristics of life. Students, I told you, you do not need to memorize these. You just need to be able to see the word and kind of describe what's going on. Some of them are really easy, like response to stimuli. It practically gives it away in the words. Other ones can be a little harder, like homeostasis. So why are the fish in our classroom, our 5 guppies, alive while the rock in there is not? Give an example of how the fish fulfill the characteristics of life listed below. So go ahead and pause the video and see if you can describe each of these and how the fish fulfill them. All right, let's see if you got it. Sorry, here was our slide on it. So for fish, well, they're definitely going to eat food. We feed them every day, and they use that food to grow, develop, repair any damage themselves, and reproduce. So they respond to their environment. Our fish respond to temperature and light. If it gets too cold, they're not going to like it. We turn the light on, they respond to it. They think food's coming. They grow and develop. So they started out as tiny little fishies, and then they grew and they developed into their adult-sized guppies. They are capable of reproduction. And this one, right, you can't see DNA within someone. I mean, we'll do a lab where we take it out of a strawberry. But we can see that, look around at your brothers and sisters, you can see that children resemble their parents, but they're not identical copies. And that's because of hereditary information encoded in DNA. So a section of DNA is called a gene and is passed on. So that our fishies can pass on their genes. That's all you have to say for that one. So, fish are made of cells. And here's one that will, I definitely want you to notice word, homeostasis. Homeo meaning the same, stasis standing still. So we're going to try to keep a stable internal environment. So whenever anything changes, your body tries to work on the change to maintain a stable environment. Good example would be temperature. If you get really cold, you start shivering. You get really hot, you start sweating to try and maintain the same internal temperature. Another good example would be glucose levels. Right? You eat a big meal, you're going to release insulin so that you take up some of this glucose in your cells. You've been fasting for a while, you haven't had anything to eat, your body's going to release glucagon so that you can break down some glycogen so that you can have glucose levels in your blood. You do the same with water and with pH. All right, students, that's it for Unit 1. So really, if you can... the big thing is if you can look at an experiment, figure out the independent variable, the dependent variable, constants or control variables in the control group, and then if you can describe the characteristics of life. One thing that might help you with the independent and dependent variables is mix dry. The manipulated independent variable goes on the x-axis. The dependent responding variable goes on the y-axis. Good luck studying, and we'll keep reviewing in class.